Hair evidence. So what information can hair provide? It is class evidence, which means that we can use it to describe a group of people if they're from an ethnicity of Asian, Caucasian, or African. It can easily be transferred to other surfaces, and this is called secondary transfer. So if your hair gets onto the couch and then somebody sits on the couch and your hair gets stuck to them, that would be a secondary transfer. So it's going to go somewhere else. And that would also relate to Locard's principle of exchange. It's also good evidence because it does not easily decompose or break down, and it retains its structure for a long period of time. And if the follicle is present, then we can get DNA possibly, and then it would become individual evidence. So if it's just torn hair or cut hair, it's going to be class evidence. But if the follicular tag is present because it's pulled out from the root, then it's possible to get DNA and individualize it. So hair's function really is to regulate body temperature, to decrease friction, and to protect against sunlight. So that's why there's hair all over the body. And you think of mammals, all mammals have hair. It is a sense organ, so it's used to detect, feeling to sense the nerves. And in many mammals, it's very dense, and then we call it fur instead of hair. We have about 5 million hair follicles when we're born, and then as we get older, we lose some of them. Let's take a look at the structure of hair. So here's a diagram, and there's two parts, the follicle and the shaft. The hair shaft is actually this part out here outside of the skin. That's what you see on your arms and your head. Now, if we look at where the follicle is, that's down here around the root. Remember, the dermis is the living part of the skin. The epidermis is that top layer, which is not living. Then we have this muscle here, the erector pili muscle, which is going to help for the hair to be able to move up and down. The sebaceous gland, you have your oil glands, and you'll also find sweat glands in the skin in that area. So the follicle is going to be club-shaped, and it's really far into the dermis. And the papilla are the blood vessels that supply nutrients that feed the hair. And the hair shaft is going to be made up of keratin, which is a very strong protein and flexible. And on the outside of the hair shaft, that's the cuticle, and that's the part that's going to be made of keratin. So if we look at the structure of the hair shaft, we can compare it to a pencil. So if you think of the outside paint on the pencil, the yellow paint, as the cuticle, that's the very thin outer layer, that's the cuticle of our hair shaft. The cortex is going to be the thicker inner part, or the wood part of the pencil. This would be our cortex in here. And then the thin, graphite, or the lead, as we term it, is inside the pencil. That's going to be the medulla. So let's look at the cuticle. And the cuticle is actually a transparent outer scale structure, which you can see here under this electron microscope picture. And it covers the hair, and it always points towards the tip end, which is good for identifying toxins, drugs, or metals. So we can actually figure out a time frame that somebody was last drugged or that they were taking certain drugs. So that's going to be formed from hardened or keratinized cells. And this picture over here is actually a human hair under the compound light microscope that we use in the classroom. You can see that cuticle pattern. Now, we have 3 different cuticle types, and this is going to help us differentiate between animal and human hair. Coronal patterns kind of looks like a stack of crowns, and we're going to find that in rodents and bats. The spinous pattern looks like spines, and we'll find that in cats, seals, and mink. And the imbricate pattern we'll find in humans, dogs, and many other animals. So the imbricate pattern is what we're going to see, and that's what we saw on the last slide here. This is the imbricate pattern up close. Let's look at the cortex, which is the largest part of the hair shaft. It's the thickest. It's going to contain pigment granules, melanin, that gives hair its color. And depending on the density and the distribution of these granules, that's what's going to help us for points of comparison if we're comparing 2 hairs under the microscope. And then the medulla is the center of the hair, and it can be hollow or filled with cells. It's a predominant feature in many animals and can contain pigment granules or be unpigmented. And hair is classified into 5 groups depending on the appearance of the medulla. And this is just for humans, so we can have a continuous, interrupted, fragmented, solid, or absent medulla. So here's some pictures of them. So this would be our continuous medulla. Interrupted, you have even breaks in between each piece. Fragmented, they're uneven spaces between the medulla. And then you have solid or none. Now, if we're going to compare animal hair and human hair, we could look at the medullary index, which measures the diameter of the medulla relative to the diameter of the hair. And animals other than humans have a much larger medullary index. For example, this is a cow hair, and we're going to have a medullary index of 0.5 or greater. So that means that this medulla is actually going to take up at least half of the total width of, or the diameter of this hair, whereas the human hair is going to be 0.33 or less, so less than a third of the diameter of the hair will be taken up by the medulla. Now, there's three developmental stages of hair growth. The first is the anagen stage, which is the active stage, and it lasts about 1,000 days. 80 to 90% of your hair is in this stage, and when it's pulled, you may actually have a follicular tag, which is important for DNA. Hair that's in the catagen stage is only 2% of your hair, and it's kind of a transition stage, and the root looks elongated, and the root bulb starts to shrink, and it gets pushed out of the follicle. And this is why we end up with grays. And the telogen phase is the last phase, and that's the hair follicle is actually dormant, so it's not growing any hair, but the root has a club-shaped appearance, and the hair is pushed out, causing shedding. So this is when hair is going to be easily lost. So some of the hairs that we find could actually be from the telogen phase. Now, there's hair classified in different parts of the body. There's 6 types: head hair, eyebrows and eyelashes, beard and mustache, underarm, body hair, and pubic hair. And they're all classified by their different characteristics. So you can see that their shape, actually, if we were to look at it up close, would be circular or elliptical for head hair, and then we'd find tapering ends on our eyebrows and eyelashes, but underarm, body, and pubic hair all are oval and triangular. So we need some more characteristics to identify them. So here we have hair that's from the arms and legs. We can find that typically the ends are blunt, and they may be frayed from abrasion, rubbing against clothes. Beard hair is usually coarse and may have a double medulla. So you'll actually see 2 medulla here. And pubic hair, there may be some buckling present, which you can see here. It's kind of this folding. Now, hair varies in shape, okay? So the cross-section may be circular, triangular, regular, flattened. The texture, it can be coarse or fine. So if you've ever rubbed up against anybody that has beard hair, it tends to be very coarse. And color, depending on the distribution of pigment granules. Remember, those granules are going to be in the cortex. Now, we also see variation amongst different ethnicities. So people of European descent tend to have generally straight or wavy hair with small and evenly distributed pigment granules and oval and round cross sections, and hair may be blonde, red, brown, or black. Asians tend to have straight hair with densely distributed pigment granules with a round and large diameter, and the shaft tends to be coarse and straight, and they have a very thick, continuous medulla. And then African hair tends to be kinky, curly, or coiled, with pigment granules still densely distributed, clumped, or they differ in size and shape. And the cross-section tends to be flattened with moderate to small diameter. Now, we can tell hair has been treated because of the different effects. So if hair has been bleached, it actually removes the pigment granules, and hair is going to tend to have a yellowish tint, and that ruins the cuticle. If we dye hair, it's actually going to change the color of the hair shaft because the color is being deposited on top of the hair, whereas bleaching is actually going to remove the color. So here's a picture of dyed hair. You can see this kind of blue color along the cuticle here, and that's because it's deposited on top. Now, animal and human hair again, we can look at the distribution of pigmentation. So humans tend to have denser distribution towards the cuticle, and animals are more towards the medulla. And they have something called ovoid bodies, which are solid masses of pigment. So to identify and compare hair, we can look at the scale structure, which would be the cuticle pattern. We can look at the medullary index, the medullary shape, and other factors if it's been dyed or bleached. And we can also measure hair because it grows about 1.3 centimeters per month, and that's just an average. Now, to collect hair evidence, we need to have numerous reference samples, and we can collect them by plucking, scraping, shaving, or even sticky tape. And when using head hair, we need at least 50 full length hairs, And if we need to collect pubic hairs, we need at least 25. And if it's a rape case, the victims have to be combed for loose hairs, and then the comb gets sent for evidence. And then to test for substances in the hair shaft, we can look for things like arsenic, lead, and drugs. And the hair is dissolved in an organic solvent to break down the keratin. And then we can establish a timeline of, knowing how about the average of how long the hair is going to grow, figure out where we find the toxin. And we can use neutron activation analysis, NAA, which can identify up to 14 different elements in just a 2 centimeter piece of hair. And that's going to use silver in order for that test to work. So there's many different things that we can use the hair for. It's a matter of being able to compare and identify and make sure it's animal hair or human hair. And then from there, you take the steps to identify it further.